Hello students, today I Swati Verma, Assistant Professor at School of Medical and Allied Sciences, Calcutta University, shall be discussing with you about restriction endonuclease. In this particular lecture, we will discuss about what are restriction endonuclease and how they will perform their action. So if we talk about your bags, that is your bacterial artificial chromosomes. So your bacterial artificial chromosomes are similar to your Escherichia coli plasmid vectors. So these are similar to Escherichia coli plasmid vectors. This is the structure of bag. They contain ORI, that is your horizon of replication, and genes which encode ORI bind F proteins. These proteins are critical for back replication. So these particular proteins, BIND, INF, BindF proteins, are critical for your back replication. It is derived from naturally occurring F plasmid. The DNA insert size will vary between 150 to 350 kb. A large DNA insert of up to 200 kb can be cloned. They are used for cloning inside your eukaryotic cells. These act as eukaryotic chromosomes inside the host eukaryotic cell. It possesses the east telomere at each particular end. A east centromere sequence, that is your CEN, is present, which allows proper segregation during meiosis. The ORI is bacterial in origin. Both yeast and bacterial cells can be used as host. Now if we talk about restriction endonucleases, then your restriction enzyme is basically your bacterial enzyme that cut DNA molecules only at the restriction sites. These are categorized into two groups based on the type of cut, how they will cut your DNA molecule. So these are cut with your sticky ends and cut with your blunt ends. So we can categorize them into two types. First, your cut with sticky ends and the cut with blunt ends. These are named after the organism from which they were derived, that is E. coli from Escherichia coli, BAM H1 from Bacillus aeruginosa. These protect bacteria from bacteriophage infection. These restrict your viral replication. Now what are the various steps? So first is the isolation of your foreign DNA. Then your PBR322 is opened. Your foreign DNA is ligated. Recombinant plasmid loses its resistance to tetracycline. Now your various classes. So these are basically divided into two classes. Class type 1 is these cuts the DNA on both strands at a non-specific location at varying distances from the particular sequence that is recognized by the sterification enzyme. Therefore, random or imprecise cuts. Not very useful for your rDNA applications. Now if we talk about your type 2, so these cut both strands of DNA within the particular sequence which is recognized by the restriction enzyme, used widely for molecular biology procedures, that is DNA sequence is your symmetrical. Now if we talk about your E. coli, these reach the same in the 5:3 direction on both strands, that is palindromic sequence. Some enzyme will generate your blunt end, that is will cut it in the middle. Other will generate your sticky end, that is your staggered cut. H bonding possible, hydrogen bonding is possible with your complementary tails. DNA ligase will covalently links the two fragments together by forming phosphodiester bonds of the phosphate sugar backbones. So these are your steps. First is your restriction enzyme will cut its slides. This is your sticky end, these are your blunt end. Now your 3'N and your 5'N, your sticky ends E. coli, that is the step of digestion, will occur. Your blunt ends digestion and then the step of ligation. So this is the formation of your recombinant DNA. Now if we talk about your DNA ligases, then your DNA ligases catalyzes the formation of phosphodiester bond between your 5' phosphate of one strand of DNA or RNA and the 3' hydroxyl of another. So your DNA ligase will catalyze the formation of phosphodiester bond between the 5' and your 3' and this enzyme is used to covalently link or ligate fragments of DNA together. The DNA ligase used in case of molecular cloning will differ in their abilities to ligate non-canonical substrate such as blunt-ended duplex DNA, RNA, hybrid or ssDNAs. Now the functioning of your DNA ligase. So your bacteriophage T4 DNA ligase, then we have E. coli DNA ligase, tag DNA ligase and T4 DNA ligase. So these are your 4 types of DNA ligases. This is your E. coli treated plasmid DNA. Then the first is the selection, then comes to your random association. These fragments will randomly associate. Then comes the step of DNA ligase. This is your calcium chloride treatment. Then comes to your bacterial colonies formation by means of transformation. Here your plasmid DNA isolation and the characterization. Now your bacteriophage T4 DNA ligase. So the most widely used DNA ligase is derived from the T4 bacteriophage. It is a monomeric polypeptide. Molecular weight is 68 kilodalton, which is encoded by bacteriophage gene 30. It has border specificity and will repair your single stranded nicks in duplex DNA, RNA or DNA RNA hybrids. So this is the functioning of your T4 DNA ligase. Now if we talk about your E. coli DNA ligase, so it is derived from E. coli cells and require NAD plus as cofactor. It is a monomeric enzyme having a molecular weight of 74 kilodalton, which catalyzes the formation of the phosphodiester bond in duplex DNA containing cohesive ends. This enzyme has narrower substrate specificity, which makes it as a useful tool in case of specific application. Now we talk about tag DNA ligase. Then the gene encoding thermolabel ligases have been identified from several thermophilic bacteria. Several of this ligase has been cloned and expressed to high levels in case of E. coli. It is used in the detection of mutation as thermostable DNA ligase, which retains their activities after exposure to high temperature for multiple rounds. It is used in DNA amplification reaction to detect mutation in mammalian DNA. Now comes to your T4 RNA ligase. So T4 RNA ligase is the only phage RNA ligase that has been extensively characterized and used in genetic engineering. This enzyme catalyzed the phosphodiester bond formation of RNA molecule with hydrolysis of ATP to PPI. So it will hydrolyze your ATP to PPI. It is a monomeric enzyme with 373 deduced amino acid residues that is a product of your T4 gene 63. Thank you.